Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com webinar titled The Variant-Rich Biobank, an Insider's Look. I'm Liam from the events team here at Scientist.com, and I'm pleased to be your host for today's event. This webinar has been sponsored by Precision for Medicine, so a big thanks to them for helping to make this event possible. And with that, I'm very pleased to welcome the first presenter, Anuj Khalsi. Anuj, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thanks, Liam, for the introduction. My name is Anuj Khalsi, and welcome everybody to this webinar for Precision titled Variant-Rich Biobank and Insider's Look. Me and my colleague, Dr. Robert Snyder, will walk you through our capabilities and an introduction to a variant-rich biobank and different services that we have surrounding those. I'm the scientific director in the Biospecimens group with over 2 decades of experience in pharma as well as in academia, focused on cell biology and immunology. And it's a pleasure to be with you today to talk a little bit about some of the company's capabilities as far as the biobanks go. So for the agenda today, we're going to start off with just a quick introduction to Precision and giving you a quick overview about it. Then we'll move over to specifically talking about the variant-rich biobank, some of the Precision's products under biospecimen services, and some of the custom solutions that we can actually provide for you and fuel and innovate your research to drug discovery further. And then my colleague, Dr. Robert Snyder, is going to connect the dots and sort of talking about a very interesting and an exciting opportunity from our company's perspective, which is called Project POSI, literally standing for Precision's Oncology Sequencing Initiative. So Precision for Medicine is a global CRO and supporting clients' biomarker-based informed R&D activities from bench to market. We have large clinical networks of biobanks providing high-quality biospecimens to our clients that really sort of support their clinical trials. We have labs across North America and Europe that support these critical R&D activities globally. We also are very privileged to support clients' data science needs as well as regulatory strategies and market access needs. As you can see on this slide, we really have a sort of a global footprint for patient samples and data collections. We have a huge biorepository with over 30 million biospecimens that are being managed alongside sort of critically supporting the research activities in 7 labs across North America and European Union, processing millions of samples to really sort of fuel the innovation for our clients. We have completed over 500 plus clinical trials for therapeutics, IVD, and diagnostic company clients. So in our experience working with clients, what we have realized is that experimentation reproducibility is a critical problem in the scientific community. And what we have figured is biospecimens are sort of the key to providing a solution to this problem. This experience of ours was sort of backed by a Nature survey in 2016, where over 70% researchers have had responded and concluded that they have tried and failed to reproduce scientific experiments of other researchers. Later on in 2018, following Nature survey, 86% of those respondents acknowledged poor reproducibility of scientific experiments in the community. So as you can clearly see, the research community sort of relies on the biobanks to deliver a fit for purpose samples. And Similar sort of a study done by Reproducibility project, as you can see on the slide in the center and to the right, sort of suggested similar statistics of poor reproducibility. And so a solution to this problem is something that Precision offers, which is a high quality biobank with extremely high quality and annotated biospecimens that can really solve this reproducibility problem, because it leads to a huge resource and time waste in the research community. As you can see in this particular slide, we have a vast array of biospecimens that are collected from thousands of appropriately consented and clinically annotated specimens that are in our biorepository. They really range from blood and biofluids and similar derivatives to tissues, various tissues that we have, which are pathologists verified. We have a huge repository of viable cells that are HLA typed, including, all sorts, include PBMCs, bone marrow mononuclear cells, leukopaks, dissociated tumor cells, and many more. We have an ample variety of liquid biopsy samples, that we have in our biobanks for fueling research, as well as a high degree of capability for doing custom biospecimen collections through our global clinical networks. And so one thing that I do want to share is a personal experience on how something like a precision biobank really sort of helped me as a researcher in pharma. And just quickly to give you the problem is, we were dealing with a breast cancer um, um, drug discovery program where we were trying to figure out the HER2 status on a particular sample type in the breast cancer arena. And having to have access to samples, tumor samples from HER2 breast cancer, HER2-driven breast cancers were kind of rare. And so at the time, my old employer had reached out to Precision to really sort of acquire these samples to get a profiling of the HER2 expression pattern in the tissue types that we had acquired at the time. So you can really sort of see how these biospecimens, especially that are well characterized and annotated, could be a critical research tool to fuel your research and innovation to drive the drug discovery forward. And there have been many such examples like these that have come across, but I wanted to kind of share with you one particular experience like such. So with that, as I mentioned, we do have huge custom collection capabilities. We are experienced global network for principal investigators and labs that are spread across, 150 plus investigator sites. We have different therapeutic areas that we're dealing with, covering over 90 plus medical conditions spread across 25 plus countries, and allowing for a huge amount of phenotypic data points that would really guide you through your research and development activities. Besides that, we have a global presence and over 500 plus actively enrolling studies that are going on with clients to help support their research needs. So with that, as you can see, not only do we have a huge array of biospecimens, but we have different capabilities all across, from discovery to really sort of the end of the pipeline, aspects in drug discovery and research. And with one such initiative, with a huge amount of biospecimens, what our company was able to do is venture into a very exciting sequencing initiative, which my colleague, Dr. Robert Snyder, will talk about, called as Project Posey. And so with that, I would like to pass the stage over to Dr. Snyder for him to educate us and share the excitement about Project Posey and some of the similar initiatives that the company has undertaken. So thank you very much, and over to you, Dr. Snyder.
Thank you, Anuj. I'm Robert Snyder, and I'm the Associate Director of Genomics. I'm really excited to be here today to talk about the Precision Oncology Sequencing Initiative that we're calling Project Posey. Just a quick overview of the data. Some of the data we've generated from patient samples. We're screening FFPE blocks at a rate of about 800 samples per month. We are ramping that up very steadily. We're going to be up to 1,600 samples per month in the coming months. And so actually, by right now, I think we're closer to 9,000 tumor cases screened. Additionally, we have ongoing prospective collections for liquid biopsy assays where we screened around 350 donors across a large range of indications. First, with tissue assay data for enrollment, and then with plasma assay data. So this is matched FFPE plasma and that we have for our biorepository. So this is some of the equipment that we're rocking right now. We've got a NextSeq. We just installed the NovaSeq platform. From Thermo Fisher, we have a lab full of Gen X's. On these instruments, we're running the Thermo Oncomine Precision Assay. It's about a 50 gene panel. We're in the process of expanding to also run the Oncomine Comprehensive Assay, which is more of a 500 gene plus MSI and TMB, along with the Illumina's TSO 500 high throughput, which is also a 500 gene panel with MSI and TMB. And in the plasma arena, we're running the Pillar OncoReveal multi-cancer assay. So I guess that the big takeaway from this is that we're really platform agnostic and assay agnostic when it comes to screening. We don't have a horse in the race, so to speak, as to who's developing what, but rather we aim to let the data speak for themselves. This is a great source of head-to-head comparisons, but it is not our intent to rate the different assays or instruments, but just to have them available for oncology insights for whatever purpose an investigator might want. In fact, I guess the real point is that if there's an assay or an instrument out there with data to be had, then we can run it. So next I'll tell you about our specific projects within Project Posey. We've got Project Spawn, which is making up about half of our projected 2022 throughput. Now this is a partnership initiative where we're sequencing the 800 FFPE samples a month on the Thermo Fisher GenXus instruments. What we've got, the numbers represented here, is what we've done already across the breakdown of indications seen using the Oncomine Precision Assay. This 50 gene panel includes SNVs, indels, fusions, and amplifications, or copy number variants. Now this is in parallel with LumaSpawn. Luma for Illumina, since it's run on a NovaSeq. We're expanding the list of indications, the genes increasing to 500 total, and increasing the different kinds of variants to include MSI and TMB. And this is a breakdown of what we estimate to run in terms of indications through the rest of 2022. Now, let me tell you about this from my personal point of view. I was working in CDX product development, all the way from early R&D, feasibility, verification, validation, into submissions for CE, PMA, 510K, and the biggest bottleneck, particularly when running accuracy assays, is samples. To prove that you can detect what you can detect, you've got to do it over and over again and provide that statistical power. So I was trying to find screen samples. But when I was developing a plasma assay, the best that I could do at the time was matched tumor. And I was absolutely all over that. But when I ran out of that, then I was just sequencing unscreened samples, as many as I could, and praying to the mutation gods that we would find those rare variants that we needed for our submission. And this was really hard, not just technically, but also emotionally, because we had so much on the line. Trying to meet deadlines and and really what I needed then was basically what I wish I had, an easily searchable database of samples and their associated variants to choose from. And ideally, along with the demographics and the metadata, as well as the raw sequencing data, um, you know, any orthogonal data would have been great. Um, but what we've got here now at Precision, it would have been a gold mine for me. I mean, it would have made accuracy so much easier. Um, so this is what I wish I had. I wish I had Precision's biorepository at my disposal back then. So, you know, the way that we're able to do such an expansive screening initiative is it's not just row sequencing, but it's through partnerships. It's win-win situations where we have the samples, we've got the infrastructure and some expertise with the wet lab work, and we generate data for our partners. CDX developers need samples and data. They provide kits for us to use. We run them, we co-own the data. They have the options to buy samples if they need them. And we get our biorepository characterized with NGS data. Um, well, this was quite a pleasure to have you guys join us today. And um, I guess there's a link in here if you'd like to contact us on LinkedIn. And with that, I will turn it over for any questions that you might have.
Excellent. Well, thanks so much, Anuj and Robert, for the really great presentations. Um, just bring you back online here. All right. All set to answer some questions?
Yeah, absolutely.
Awesome. Well, Anuj, I'll direct the first question to you here. So you were talking about viable cells in your cellular product offerings. What are your current leukopak offerings, and are you expecting to make other ones available?
Sure, thanks, Liam. That's a good question. So currently we offer a huge degree of leukopaks that we collect excuse me, internally in our FDA registered blood center and apheresis center. So we're able to offer healthy leukopaks that are full packs that come with a guarantee of 8 to 10 billion cells, although our leukopaks have been sort of even much higher in quality where we clients are usually seeing about 12 to 15 billion cells. The volume on these packs is anywhere between 240 to 300 ml. And for the half packs in the leukopak category, there's about 4 to 6 billion cells with a volume of about 100 to 140 mils. We also offer disease state leukopaks, which are half a pack. Anywhere between 3 and 5 billion cells, also within that 100 to 140 mil volume. We, as I mentioned, we definitely have a FDA-registered blood and apheresis donor center, along with many of our partnered network collection centers that are able to fulfill these for us, for our clients. We have multiple trained nurses as well as phlebotomists, and we characterize these samples. They are well annotated. We also, don't forget, have the opportunity to do custom collections, and I really encourage you to reach out for any of the needs for custom collections through over 150 plus investigator sites, really sort of deeply characterized and phenotypic data that we have available on well QC'd out samples. The other part to that in the leukopaks, so we're very excited to be coming up with and offering sort of these new product offerings called the cryopreserved leukopaks in Q2. And these will just sort of allow the researchers the flexibility of using that same fresh leukopak, but at the ease and the convenience of their own time to drive sort of research. And so those are going to be highly qualified as well, and will be available for offerings very soon. So I encourage everybody here to sort of be on a lookout, and please reach out to us and keep us in mind. And the third part to that is towards the third quarter to the end of the year, we're also looking to offer some mobilized leukopaks. And the reason that these are important for research is because these are highly driven CD34 hematopoietic stem cell rich leukopaks, and we're very excited about this product. So again, encouraging everybody on the call to be on a lookout for this. Please reach out to us and help us sort of partner in with your research needs to help you kind of deliver that first-in-class, best-in-class drug to the patient's needs.
Fantastic, great answer, Anuj. All right, Robert, I'll direct this next question to you. You were saying out there that your platform is agnostic, and if there's any sequencer out there, you could run it. Is this really the case? So, you specifically mentioned Thermo and Illumina, but could people get your data from samples generated when on one of the newer companies' sequencers out there?
I did say that, didn't I? Putting me on the spot. The short answer is yes. The longer answer is let's talk about what you're trying to do and how we can work together. I know that there are a couple of newly released sequencers. There's Singular Genomics and Element Biosciences. And I also heard that Oxford Nanopore Technologies is entering the diagnostic space, and all of these companies are partnering with some big name CDX companies out there. So it shouldn't be too long before these sequencers are going to be a viable, if not like preferable platform to your Thermos and your Illuminas, particularly for like your smaller fit for purpose clinical lab space. So what I expect to happen is, as these technologies mature in the marketplace, there will be in parallel an increase in demand from CDX developers to get data generated from these platforms on real samples. So if that is something that you are pursuing, then definitely hit me up, because I would love nothing more, and it's part of my remit to keep precision at the cutting edge of the state of the art.
Excellent. Yeah, another great answer. So here's a good one here, and I'm not sure if who best to direct this to, but do you have blood samples that are available?
I can take that one, Liam. Yes, we do have blood samples that are available. Whole blood samples are available from healthy donors for control purposes to covering sort of multiple different therapeutic areas and different disease conditions. As I mentioned, we have over 90 plus medical conditions that we really sort of cover across the board. And so we do offer blood PBMCs, including plasma and other related products. And so I really encourage you to reach out to us and discuss with us your specific therapeutic focus area. And if there is a disease type that you're looking at, that you can reach out to us, and we'd be very happy to help you provide research solutions that you might be looking for. Thanks.
Perfect. Yeah, thanks, Anuj. And just want to point out that Anuj's and Robert's emails are on the screen. If you do want to reach out, you can click that button there on the screen, and it'll pop up in an email. All right, next question here, and Robert, maybe you can lead this one. Can we get a few samples with known variants to validate an assay in our lab?
Yeah, definitely, and that's what we do. But if you're interested in validation, we can build the kits for you. We can even, we could put those samples on reserve, and then if you need to validate again or send out to different labs, we can have basically pre-built kits ready to go on demand. And we can help if you've got your validation plan already together. That's great. If you need help putting together a validation plan, we can help with that too. And we can even, if you need to do things like serial dilutions of variant allele frequency for limit of detection, we can do that in our lab and have all the samples blinded, have a unblinding key ready to go. So yeah, like validation kit building is definitely in our wheelhouse.
Awesome. And I think in the interest of time, we'll just have one more question. And Anuj, I think I'll direct this one to you. So they said, they're excited to see DTCs being offered by Precision. Can you expand a bit on this product line and maybe share any details with us?
Absolutely, Liam. That's a good one. Thank you again. DTC is really standing for dissociated tumor cells. Very exciting product line. We have multiple different tumor types that we offer. Primary processed DTCs that are P0, passage zero. And essentially, the reason that these are such great research tools is because they sort of capture the heterogeneity of the cellular samples that is present in the tumor type that you're looking to investigate. And so we have a breadth of DTCs. We're also continuously looking to expand our inventory. So I encourage everybody to sort of just reach out for your dissociated tumor cell needs. What other thing that we have and offer besides DTCs is actually matched PBMCs as well as plasma sets. So you can really sort of do multitude of assays, cell-based assays, analyzing analytes of different kinds alongside of looking at these primary cells that contain all different cell types that might be present in a tumor environment, including TILs, for example, tumor infiltrating lymphocytes, a stroma that's associated with tumor, the epithelial and the endothelial cells that might be in there. So there's a whole and cancer associated fibroblasts. So there's a whole degree of cell types that get encompassed in the DTCs. So really an exciting product line. I encourage our viewers to reach out to us and take the advantage of these products and really help drive your research further by not only utilizing the DTCs but even the matched PBMCs and the plasma sets.
Fantastic. Yeah, great. Perfect. Thanks so much, Anuj and Robert, for sharing your insights here with us today. It's been a real pleasure having you with us.
Absolutely, it's our pleasure.
Thank you. Likewise.
Yeah, and a big thanks also to the audience here for participating. And last but not least, of course, we'd like to thank Precision for Medicine for sponsoring the event. So in closing, we hope you enjoyed this scientist.com webinar, and we'll see you again next time. Have a great day, everyone.